Thanks everyone for watching my presentation. I'm Qinyu from Brigham Young University working with Dr. Douglas Tree. So today I'm going to talk about some recent progress in our group about understanding the shape manipulation of amphiphile aggregates in solution. So the motivation of our research originates from the concept of morphogenesis. It's a biological process that describes the behavior of organisms taking forms and changing shapes at different length scales and levels. It would be rewarding to replicate such behaviors in a synthetic system so that we could apply it to different research areas. For example, we can make protocells that can divide and replicate for self-repair, or synthesize artificial tissues for medical treatment, or at an even larger scale, create certain patents and use it for controlling mini robots. However, this biological process is also complicated and involves various regulatory factors and requires a lot of information exchange routes. Imagine if we want to study all the factors exhaustively, we would need to perform a huge amount of experiments or simulations. So it's natural to first consider understanding a simplified system. Among all the potential candidates, responsive block copolymer vesicle is of particular interest because it has a similar basic structure as the biological cell membranes and also demonstrates some interesting behavior when exposed to specific stimuli. Most commonly used ones include light, temperature, and pH or other concentration gradients. If we can keep the stimuli in a localized position, we might be able to see localized deformation or spatial migration of the vesicles. So in today's talk, we will focus on studying the localized shape change induced by a concentration gradient. We used a minimal model consists of a generic block copolymer vesicle with incorporated enzyme or externally introduced stimuli to give rise to certain chemical reactions, and eventually the reaction would result in some local deformation. So to simulate such a system, we would need a coupled reaction diffusion model that can simulate the motion of particles as well as the dynamic changes during chemical reactions in a polymer solution. Accordingly, we developed this reactive DPD model by combining dissipative particle dynamics and split reactive Brownian dynamics. So DPD is a coarse-grained model with pairwise interaction between particles, and it also includes the hydrodynamic interactions that are important in vesicle formation. The basic DPD model includes three different forces. The conservative force controls the miscibility between different species. The dissipative force accounts for the drag of fluids, and the random force describes the thermal fluctuation of the system. And for polymer chains, we added a harmonic bonded interaction between neighboring beads in a single chain. SRBD model is an efficient event-driven model to simulate reactions. It assumes that when two particles come into contact, there's a possibility that the collision will trigger a reaction. So more details could be found in the references. So with the developed model, we first observed local swelling driven by embedded enzyme particles that catalyze the conversion of surrounding solvents into another solvent, which is shown in red, that interacts favorably with the solvophobic blocks. The figures show a time sequence of the simulation in which we can observe a clear local swelling compared to the initial vesicle shape because the converted solvents tend to diffuse in and mix with the solvophobic blocks, and the extent of deformation increases with the simulation time due to the rapid production of solvents. We then explored another mechanism which introduces external stimulus particles, which is shown in orange, to convert the solvophobic beads into solvophilic ones. The pink particles represent the converted solvophobic particles. As we can see from the figures and video, this change also results in a similar local deformation to the solvent swelling case, but in this case, instead of having solvents diffusing in, the converted particles tend to diffuse outward and stretch into the surroundings. So despite the apparent similarities between these two different cases, we still see some major difference when testing the long-time behavior. We performed fast kinetic simulations for both cases, where we either introduced a droplet of good solvent for the solvophobic blocks near the vesicle or instantaneously changed the block fraction of part of the chains in the outer corona of the vesicle. We ran longer simulations to see the full range of dynamics. The end of simulation snapshots suggest that in the solvent swelling case, the solvents distributed evenly throughout the whole vesicle, and the vesicle returns to a spherical shape with a slightly increased size. Whereas by changing part of the chain's block fraction, the vesicle still shows a slightly elongated shape where the block fractions were altered, even at t equals 25,000. So this suggests that the latter mechanism could provide a more long-lasting local shape change compared to the solvent swelling mechanism. We got some further insights by comparing the diffusivities of the polymer chain and the solvent bead. The figure on the left shows the mean square displacement as a function of time. The fitted diffusivity of polymer chain is about 15 times lower than that of the solvent particle. The videos on the right might give you a more straightforward demonstration of such difference, while the single particle, which represents the solvent bead, has diffused across the periodic boundary for a couple of times. The polymer chain is still like wiggling around its initial position. Therefore, we think that the relatively longer diffusion time of polymer chains result in the more persistent local deformation. So to summarize the work in our recent publication, we used a reactive DPD model to investigate the morphology change of polymer vesicle due to either solvent swelling or changing the polymer properties. Our results suggest that changing polymer properties would render a more persistent local shape change, which we think is due to the longer diffusion timescale of the polymer chain relative to the non-bonded solvent particles. So far, our work is based on generic diblock copolymer model with simple repulsive potential. Moving forward, we would like to explore a more realistic system. Our collaborator has been using a pH-responsive surfactant called MSDH to create a protocell system. The head group of MSDH can be protonated or deprotonated under different pH conditions. So our plan is to use Martini 3 force field instead of the solved repulsive potential to model their system. Martini 3 is a coarse-grained MD model that maps a series of Lennard-Jones parameters to specific chemical species and groups. We chose this force field because it's more sophisticated and agrees very well with the experimental results. So before moving on to the reaction kinetics simulations, we want to better understand the phase behavior of the system. So we constructed this phase diagram of the system as a function of concentration and the fraction of protonated MSDH, or in other words, pH of the system. So we can roughly divide the phase diagram into 4 different regions. In general, from top to bottom, we can see a transition from micelle to vesicle when we increase the pH. And from left to right, if we increase the concentration, the structures aggregate together to form larger clusters, and it's more obvious at lower pH. So recently we got some new experimental results from our collaborator. It is known that MSDH molecules form micelles at low pH and vesicles at high pH, but what happens in between has never been explored. So when they try to use catalytic reactions to continuously change the pH of the solution, they saw this huge peak in the size change, which has never been observed before. So when comparing to our phase diagram, we do observe a trend of an increase followed by a slight decrease in the size of aggregations at a given concentration, but the size difference is just not as much as what they have observed in experiments. And there might be some kinetic effects that's not accounted for between the equilibrium states, which is the reason why we want to perform reaction kinetics simulations in the future. So with that, I would like to thank our funding support from BYU College of Engineering and Simmons Research Endowment, and the technical support from BYU Office of Computing. And special thanks were given to our wonderful group. And I'm willing to take any questions right now. Thank you for your attention.